So here we're going to talk about the sense of sight, and we've got the visual receptors that are found in the eye. But before we talk about the structure of the eye, we want to talk about the accessory organs. So these are organs that help the eye in its function. Those are the upper and lower eyelids and the eyelashes. Those provide protection. The lacrimal apparatus produces tears. And then we've got the extrinsic eye muscles, and those provide eye movement. So first, the lacrimal apparatus. And the lacrimal gland is found all the way on the lateral part of the eye. It'll secrete tears. Those tears are going to be distributed by the eyelids onto the eye, and then they have to be drained. The drainage system is on the medial part of the eye, so we have these superior and inferior canaliculi. They're going to drain tears into this lacrimal sac. The lacrimal sac will then take the tears and drain them through the nasolacrimal duct. And the nasolacrimal duct actually opens into the nasal cavity. And that's why when we cry, we need to blow our nose. Tears have lysozymes, and lysozymes are antibacterial, and they help in killing some germs. For the extrinsic eye muscles, again, those are muscles that are attached, all of them attached to the sclera of the eye, which is the white portion, and they allow for movement of the orbit. There are 4 recti muscles, so superior, inferior, medial, and lateral rectus muscles. And then 2 obliques, there's a superior oblique and an inferior oblique muscle. For the actual structure of the eye, well, it's a hollow spherical organ that's made out of 3 different layers. The outer layer is fibrous, the middle layer is mainly vascular, and the inner layer is nervous. They're known as tunics. So a tunic is basically just a layer of the eye. The outer fibrous layer is made out of the cornea, and that's the anterior 1/6 of the outer tunic. It's the transparent portion, how these light rays are going to enter into the eye. It does help a little bit in focusing those light rays, and it transmits and refracts light. Refraction means to bend those light waves, those light rays. The sclera, that's the white opaque part. It makes the majority of the outer tunic. It's protective, so it's made out of connective tissue. And this is, again, where the extrinsic muscles are going to attach to. They are pierced in the back by the optic nerve and blood vessels. The middle tunic is made out of the choroid, ciliary body, and the iris. And I really want to get to a picture to explain all of these different parts. So again, your cornea is, I'm going to go through the outer tunic first. So the cornea, that's the anterior portion of the outer tunic. It's transparent. Light rays get to go in. They sometimes bend in order to gather them at the point of focus. And then there's the sclera, and that is the white part of the eye. It makes the majority of the outer tunic. And at the very back, you could see it here being perforated by the optic nerve and then the blood vessels as well. You can see the attachments of these extraocular muscles, again, are attached to the sclera. Now the middle layer, that's the choroid coat. Choroid coat has lots of melanocytes, and those are going to absorb any extra light getting into the eye. So the inside of the eye has to be pitch black. And in order to do so, we have to absorb, melanocytes are going to absorb any extra light getting in there. We've got the ciliary body. Ciliary body, it makes an aqueous humor, which is some kind of watery material or watery substance, a watery liquid that is found in this chamber right here. And it has the attachments of these ciliary ligaments or these suspensory ligaments. The suspensory ligaments help in keeping the lens in its position. There's an iris, and the iris is the color part of the eye. Okay, we all have different colors, eye colors, and it's because of the different pigments that are found within the iris. The iris also has smooth muscle fibers that can either constrict the pupil or dilate the pupil. Now the retina, that's the innermost layer, and that's where you'll find the receptors for vision, whether rods and cones. The retina is kept in its position with a gelatinous material that fills this whole cavity. That gelatinous material is known as the vitreous humor. Again, it literally plasters the retina against the choroid tunic. If there's any damage to the eye where the vitreous humor is, for whatever reason, escapes the eye, that will lead to retinal damage, retinal detachment. Now the lens divides the eye into a posterior cavity, and as we said, the posterior cavity is full of the vitreous humor, and an anterior cavity, and that anterior cavity is full of the watery liquid that we talked about, known as the aqueous humor, again made by that ciliary body. So here we'll talk about the anterior portion of the eye that is found between the lens and the cornea. In the middle of it, you will find the iris, and the iris divides it into anterior chamber between the iris and the cornea, and a posterior chamber, and that's between the vitreous humor, or between the lens and the iris. You can also see here the lens very clearly. It is a transparent biconvex lens. It is held in its place by the suspensory ligaments that are attached to the ciliary body. Here you can see better the ciliary body, where you have that ciliary process and the ciliary muscles. So these muscles are going to either contract or relax in order to change the shape of the lens in order to help us in focusing to be able to see pictures well. The aqueous humor, again, that's the water that fills in that anterior cavity of the eye. It's made by the ciliary body, and then it will circulate through the through the posterior chamber, then through the through the pupil, and then into the anterior chamber, and then it will drain through that canal known as a venous canal, or the venous canal of Schlemm. And any increase in the amount of water that is being made, or any blockage in its drainage, can lead to an increase of intraocular pressure, which is the increase in the pressure of the eye, and that's a condition known as glaucoma. Now for the ciliary body and lens, while the ciliary muscles can contract and relax in order to remove the lens, the suspensory ligaments are going to move with it because they are attached to both. And you can see a little video here. Watch that little video, and it shows you exactly how that works. Now for accommodation, we need to accommodate to near vision. So when you're looking at something close, the light rays coming from them are going way far apart from each other. They are not parallel. So we'd have to thicken the lens a little bit to increase its refractory power and to make it more convex. So you can see it here where the ciliary muscles contract, okay, and then that will lead to the relaxation of these suspensory ligaments, and the lens will become thickened. And that again is going to help in accommodation to near objects. When we're looking at something from far away, the ciliary muscles can relax, the suspensory ligaments are going to be pulled back, and the lens is going to become thin, which is actually its normal, usual shape. Now the iris, as we mentioned, it does have some melanocytes, and hence, the different colors, and it also has smooth muscle fibers. Those muscle fibers are arranged either in concentric circles. Those are known as your pupillary constrictor muscle. When they contract, they constrict the pupil. And we also have these radial muscles, and those are known as the pupil dilator muscle. When they contract, they will dilate the pupil. Okay, so the constrictor and the dilator are under the autonomic nervous system control. So if we need to dilate the muscle fibers, or it's like in dim light, okay, that will be under the control of the sympathetic nervous system. If we want in bright light, where there's too much light coming in and we want to constrict them, we can do so under the control of the parasympathetic nervous system. Now the inner or the nervous tunic, that's the retina, and it has the visual receptors or the photoreceptors. It has the rods and the cones. It also has two important spots, and those are the macula, lutea, and the fovea centralis. So I'm going to show you the image here. The macula and the fovea. Fovea, that is where you want the perfect picture to focus on there. Okay, so if you're looking at something, the picture, let's say you're looking at the clock on a wall, that clock's image is going to be right on the fovea. While everything else, that other field of vision will be picked up by the rest of the retina. So your fovea is that perfect picture, most detailed, most colorful, and fovea only actually has cones. It does not have rods. The macula, that's the surrounding area. It's a yellowish spot found in the retina. You can see it here, surrounding the fovea. So the fovea is the darker part in the middle, and the surrounding area is the macula. This part is known as the blind spot. Okay, so this is a real image. This is kind of a representation of what's going on. So you can see here in that blind spot, there are no receptors, and that's because the optic nerve is either leaving the eye or these blood vessels are exiting and entering the eye as well. And again, you could see that here where you can actually see the blood vessels, and the optic nerve would be exiting from this point. And that's why, again, it's known as a blind spot, where we do not have any rods or cones in that area. It's called the optic disc or the blind spot. For the posterior cavity, okay, which is this, anything behind the lens is your posterior cavity. Again, it's full of that gelatinous material known as the vitreous humor. It'll support the internal structure, so it really keeps the shape of the eye. And again, if anything happens to it, that would lead to retinal detachment. Light refraction is when we have to bend the light rays. So when light rays are coming at us, they'll pass through the cornea. Cornea can refract them a little bit, but it's really up to the lens to make sure that we focus the image perfectly on the fovea. Okay, so refraction is, generally speaking, is the bending of light that occurs when those light rays pass between mediums. So from the air to the inside of the eye that now has liquid. So that focusing is going to bend the light so that the image falls onto the fovea centralis. Now here are two different kinds of lenses. This is the kind of lens that we have in the eye, the convex lens, where light rays coming in are going to converge onto a point. Again, that point should be the fovea. These are other kind of lenses, the concave lenses. Those are going to diverge or separate the waves from each other. This is not the kind of lens that we have in the eye, but we can use them as eyeglasses for individuals that need them. So for focusing on the retina, you can see here this big object, okay, the rays coming in the middle, that red ray, really nothing happens to it, and it's still focusing on the fovea. But the ones coming from either sides, those are coming at an angle, and you want to refract them or bend them. So your first refractory surface would be the cornea. But again, you really cannot adjust the power of the cornea. Your second refractory surface would be the lens, and that is the adjustable one. So we can adjust that and make sure that the picture of the tree lies onto the fovea. Something that you want to notice here is that things on the retina are upside down, but it'll be up to the visual pathway and the visual cortex to reverse that image. Refraction disorders, this is what happens if our eyes do not focus the rays in the fovea. So if they focus them in a point in front of the fovea, that is known as myopia. Or if they focus them at a point far away, like behind the fovea, that is known as hyperopia. Okay, where the eye, in myopia, you will give a patient a lens that is going to diverge the rays further away from each other. So to kind of make sure that instead of them meeting here, they are going to meet at the fovea. So they will use concave lenses for correction. While patients of hyperopia, they will get a convex lens, so that now instead of them focusing the rays at a point behind the fovea, they're now pulled so that the point of focus is actually on the fovea. Myopia is also known as nearsightedness. Okay, so the patient can't see, can easily read a book, but won't be able to see maybe street signs. While hypermetropia is the opposite. That is farsightedness. They will be able to see the street signs without any problem, but it is hard for them to read. Now the photoreceptors, those are neurons, modified neurons found in the retina. They can detect light. We've got rods and cones. Rods, they're actually rod in shape. They have a light-sensitive pigment called rhodopsin, and they're much more sensitive than cones are. So they will detect any dim light. Those are the receptors that we use in the dark and dim light. They will not produce an image that is very clear. It's kind of like a shady image, more kind of, grayish kind of in color, just shades of gray that you're seeing. You have an outline of an object without any detail. The cones, on the other hand, they are cone shaped. They have different kinds of pigments. So erythrolab or erythropigment detects reds. Chloro will detect greens, and cyano will detect blues. Those, again, are less sensitive than rods. They produce colorful images, very sharp, and you will find that, again, the fovea centralis only has cones, while the fovea lacks rods. Here you can see an image of the rods and cones. So these are your rods, those are your cones, and again, they will have the pigment so that when they are exposed to light, those pigments change the structure, and that leads to the production of an action potential. Again, our visual pigments, they decompose or change in structure upon absorption of light. Rhodopsin, that is the light-sensitive pigment in rods. Again, it is much more sensitive to light than cones are, and that's why they are activated in dim light. The cones have what are known as iodopsins, so all three of those collectively are called iodopsins or conopsins. The erythrolab responds to red lights, chloro responds to green, and cyano responds to blue. And notice that it's not just like one shade of red. It responds to all of the different shades of red. And that's why I keep on saying responds to red lights, because it is more of, a whole range of reddish colors that it will respond to. But all the different colors that we see are really a mix and match of stimulating different receptors at different degrees. Now, stereoscopic vision is the ability for us to see in 3D. So we do not see things as flat, but we can see things where we have like a distance, a depth to the images, and that is your 3D image. We have stereoscopic vision because we see things with two eyes. If you cover one eye, that you are now losing your stereoscopic vision. You can still see, but you've lost that depth and the 3D image. Okay, it's because of the overlap of the two images, the one that you get from the left eye, plus the image that you get on the right eye, and them overlapping on each other, that will give you that 3D or stereoscopic vision. Now for the visual pathway, well, obviously from the retina, and then the optic nerve, that was your cranial nerve number 2. And then some of the fibers cross over to the opposite side, and that is where they cross over is known as the optic chiasm. And then we have the optic tract, and just like any other sense, it would have to go to the thalamus. The nucleus in the thalamus for vision is known as the lateral geniculate body, or the LGB, in the thalamus. And then we have the optic radiation that will take the vision into the occipital cortex, or the visual cortex in the occipital lobe. It's the optic radiation that will flip the picture back upside down. Or actually, I guess it would be to flip it right ways. So remember when we were looking at the retina, we see images upside down, and then I mentioned that it's up to the visual pathway to make sure that when we see things, we don't see them upside down, we see them in their correct positions. What are the lifespan changes that happen to senses, generally speaking? Well, for hearing loss, there is age-related hearing loss. That's due to damage of hair cells in the spiral organ, degeneration of the neural pathways, and then there's tinnitus. And tinnitus is that sense of a humming voice that occurs in the ear, whether there's a sound or not. There are age-related problems related to vision, for example, dry eye. So there's damage to the lacrimal apparatus. I shouldn't say damage, but it's aging to the lacrimal apparatus where it is no longer producing enough tears. We can get floaters. And floaters, those are crystallizations that happen in the vitreous humor. And they're called floaters because sometimes they are in the field of vision and the patient can see them. Sometimes they are not in their field of vision and they disappear. Okay, so they are literally floating within the vitreous humor. There are calcifications that happen in there, and hence the name floaters. As we age, we tend to lose the elasticity of the lens. Okay, so we lose that accommodation function, and that is known as presbyopia. That's when patients are going to need reading glasses. Glaucoma is the increase in the intraocular pressure of the eye, and we kind of already mentioned that. Cataracts are the loss of the opacity of the loss of the transparency of the lens. So as we age, or as you guys know, that a lens normally is transparent. Okay, it's made out of crystals, but then made out of different proteins, then as we age, these proteins are going to calcify. Okay, that calcification process is going to let us lose that transparency. So the lenses are going to become opaque. And if you look into the eyes of a patient that has cataract, you can actually see the lens. So there's like that cloud that you see in the eye. That would be a cataract eye. Macular degeneration, that is the degeneration of the macula. And remember, the macula is the part surrounding the fovea. And when you lose that part, when that degenerates, that can lead to loss of vision. Now, what are the age-related problems due to smell and taste? Well, taste buds really do not change, but it is the olfactory receptors that we lose as we age, leading to anosmia, which is the loss of smell, the inability to smell. And it's because of that where things tend to change, the palates start to change because they are no longer tasting food the same way. Again, no problem with the taste buds, but all of the problems are happening in the olfactory receptors. And this ends this chapter.